Okay, so best practices for biostatisticians in clinical trials. What I've tried to do here is condense the material that I teach in one semester course in our department into one presentation, although obviously I have to very carefully pick and choose the things I thought were most appropriate. A lot of what I want to talk about is kind of challenging what is oftentimes conventional wisdom. So the ideas in this presentation are all mine, do not necessarily represent the views of the broader community.
Good afternoon, everyone. My name is Teresa Linz, and I work in ICTR as a program manager. I'm really pleased to have Dr. Tom Cook join us today and present on biostatistics for clinical trials. Dr. Cook is a CHS professor in the Department of Biostatistics and Medical Informatics. For over 30 years, as part of the clinical trials program within BMI, he has helped to support independent data monitoring committees, primarily for industry-sponsored clinical trials in a host of disease and therapeutic areas. He has also served as a member of many IDMCs for both industry and NIH-sponsored trials, and is currently co-chair of the ICTR DMC for local UW investigators. With founding BMI chair Dave DeBetz, he co-authored and edited the textbook Introduction to Statistical Methods for Clinical Trials, and has been the corresponding graduate level course within BMI for over 15 years. So again, we're very pleased to welcome Dr. Cook today.
So I should share my screen. Let's see if I can do this again. Okay, everybody see my screen?
Yes, we can. Thank you.
Okay, so best practices for biostatisticians in clinical trials. What I've tried to do here is condense the material that I teach in one semester course in our department into one presentation, although obviously I have to very carefully pick and choose the things I thought were most appropriate. A lot of what I want to talk about is kind of challenging what is oftentimes conventional wisdom. So the ideas in this presentation are all mine, do not necessarily represent the views of the broader community, who might object to some of my opinions. I'm going to focus on randomized controlled trials, so early phase non-randomized pilot trials won't be discussed. And because of the interest of both the interest of time and the audience, the content will be presented a la Kenny Rogers, who said, you got to know when to fold them, or when to hold them, when to fold them. And the problem with that song is the gambler never tells the narrator when he should do any of those things. So it's not very helpful, except for the bit about never counting your money at the table. So my focus is more about how to think rather than what to think or to do in any given situation. I'll also assume that biostatisticians are trained in how to actually implement a wide variety of procedures. So I want to focus on the interpretation and the appropriateness. So by brief outline, three kind of major areas: causal thinking, and I'll mention what I mean by that in a moment. What's the question? And then I want to talk a little bit about missing data, which, as you'll see, is to me the single most important issue that we face in randomized trials. So here's the big picture. I imagine that we have data and computation and statistics. Those are tools that we use to draw inference, but the ultimate goal is for those things to tell us information about the physical universe. And the statistician who is involved in doing the data analysis and computation shouldn't simply be a programmer who does what the investigators want, but they should rather play an integral role in formulating the study design, analysis, and the interpretation of the results. And I'm also viewing the statistician as a kind of skeptic who won't allow interpretations of the results of the data analysis to be not founded in sound principles. And so this little skeptic guy, this is actually a logo that was created by a student of mine about 10 years ago. Yeah, about 10 years ago. And I've kept it, and he somehow imagined that this thing looks like me, and the little wry smile indicates that I'm being skeptical of the claims that some people want to make. So causal thinking. Clinical trials are causal instruments in the sense that their purpose is to identify causal relationships between interventions under study and the outcomes of interest. It's very rare when I read papers on methods for clinical trials that the word cause or causal or some version ever comes up. In fact, if someone sends me a paper, the first thing I'll do almost every time is to do just a search, and that's the advantage of doing everything digitally. I can search for words, so I look for the use of the word cause or causal or causation, and they're very rarely found. So, in my opinion, it's a need in our community to learn how to think causally rather than simply think correlationally. So sound RCT practices requires that we use causal thinking in order to draw correct causal conclusions. So here's one of my favorite XKCD cartoons. I used to think correlation implied causation. Then I took a statistics class. Now I don't. Oh, sounds like the class helped. Well, maybe. One, I think it's humorous, but there's actually some depth in this particular comic. So the speaker might actually realize that he has no formal basis for claiming causation. So if he took the right class, he would realize, in fact, that simply taking the class isn't enough. Or he may have actually heard or read something independent of the class that changed his thinking. So we don't really know if the fact that the class happened and his thinking changed is correlation or if it's actually causation. The other thing I've noted, the speaker would not be me, because in all of the statistics classes, which were many in my lifetime, I don't remember the question of causation ever even being discussed. So I wouldn't have taken a class in which this question would have even arisen. So in my opinion, we haven't trained statisticians very well in causal thinking. Although given that it's been close to 40 years since I've taken any statistics classes, maybe the world has changed and we think more causally than we used to. There's certainly in the last 30 years has arisen a lot of thinking and study of what I would call the field of causal inference. But almost all of what we call causal inference in the statistics universe or the machine learning universe is in observational studies and not in randomized trials where we're relying on the randomization to allow us to make causal claims. So Dave Demetz and I back, what is this now, 7 years ago, because we actually wrote the paper in 2018, we claim without justification that if we have two groups defined by treatments A and B, and we see a difference between these groups, that difference must either be caused by chance, that is, and that we can assess via statistical analysis, or it's causal, that is, B has a causal effect on the outcome relative to A, or it's due to confounding, which I define to mean systematic differences between groups that exist at the time the conditions are imposed. And then we say, because randomized assignments cannot be associated with participant characteristics, randomization eliminates the third possibility, enabling direct assessment of potential causal relationships, provided that the study is designed, conducted, and analyzed properly. So I like to say that randomization is the magic dust that we sprinkle on our data that turns correlation into causation. So when I say confounding, I said this refers to systematic differences between treatment groups at the initiation of the treatment, usually the result of some observed or unobserved factors C that are associated with both the treatment and the outcomes. And they induce a mismatch between the observed associations and the underlying causal relationships. So here's just a picture that illustrates the C represents causal potential confounding factors, could be associated with treatment, they could be associated with outcome, and then we have some association, causal association between treatment and outcome. But what we might observe when these confounding factors are present is an association between treatment and outcome that's not causal, it's induced by this confounding. And the key fact here is that statistical analysis alone cannot tell us the direction of the arrows in this diagram. We need randomization, we need designed experiments in order to be able to allow statistics to actually inform us about causal relationships. So randomization prevents the systematic association between C and T, and therefore it eliminates confounding. So if we can rule out chance via statistical analysis, then any observed associations that we have between treatment and outcome must be causal. So here's my favorite example of confounding. This comes from a paper that was published almost 30 years ago. I think I've used this example in every course I've taught in the last 30 years. So we have 1,300 women from Wickham in the UK, it's right near Newcastle, enrolled in the early 70s. They were followed for 20 years, and then it was noted at the end of that 20 years that smokers had a 24% mortality rate and non-smokers had a 31% mortality rate. This corresponds to an odds ratio of 0.68, and the confidence interval is well below 1. So if we can assume that these subjects represent a random sample from the desired population. Now inference, this confidence interval actually relies on the fact that these subjects are randomly selected. So statistical analysis doesn't simply happen in a vacuum. We have to have some basis for actually drawing statistical conclusions. So I'm going to assume that this is a random sample from the desired population, and therefore this confidence interval actually does reflect a 95% confidence interval. In that case, we have a high degree of statistical confidence that there is a strong negative association between smoking and mortality risk. It's a fact. If I pick a random woman from this time frame in Wickham, if they're a smoker, they have a lower risk of death in the next 20 years than did a non-smoker. However, I cannot, from that association, infer that it's causal. So then if we look deeper, we stratify by age, we look at the younger women up through age 64, we look at the older women. We see in the younger women that the observed death rates are higher among smokers than non-smokers. So here the odds ratio is bigger than 1. It's flipped from being less than 1 to being greater than 1. And of course, in the older women, the vast majority of them died in the intervening years, and the mortality rates are about the same. So the older women in this study were far less likely to smoke than the younger women. I imagine that's because they came of age back in the beginning of the 20th century when women were far less likely to smoke, and then the younger women came along at a time when smoking among women was much more common. And if you don't start smoking when you're young, I'm guessing as a non-smoker that you probably never start. So there's they also note that of the 190 or 39 deaths among the smokers, 42 were in the older age category, whereas that's about 30%, whereas in the non-smokers, about 72% were in the older age category, up among those who died. So age is clearly the confounder for the causal association between smoking and death. And I would add that confounding only makes sense in the context of causation. It doesn't make sense to talk about causation unless we are trying to draw causal conclusions. I reviewed a paper once where someone used the word confounder in the context of an observational study where they were simply doing prediction, and it didn't make sense in that context. However, in neither case do we have a formal basis for actually making causal claims. So just because among the younger women we see higher smoking rate or higher death rate among the smokers, that doesn't imply that smoking in fact causes an increase in mortality risk. So that's what confounding is about. Another myth, it's commonly believed that the purpose of randomization is to achieve balance between treatment groups with respect to important characteristics. But there was nothing in what I've just described that discusses balance. Randomization works because it prevents confounding, not because it achieves balance. If we have chance imbalances, these are automatically accounted for by the statistical analysis. Chance imbalances are simply one source of randomization and not sources of bias. All inferential statistical analyses are based on long-run behavior, not individual instances. So you can't look at a study and say, oh, look, we have this imbalance with respect to some factor among the treatment groups, and therefore that has somehow biased our causal inference. No, The causal inference is not based upon single instances, it's based on long-run behavior. And it's impossible statistically to determine if any given instance represents a type 1 or a type 2 error. And then finally, p-values for baseline differences are meaningless. And fortunately, I see in most journals these days, they don't put p-values in their baseline tables. That used to be common. I don't think it happens anymore. The p-values constitute a test of the null hypothesis. There's no systematic difference between groups. But we know a priori that based on the randomization, this null hypothesis is automatically true. So we're testing the hypothesis that we already know is true. So that's pointless. And then what I've done here is I've simulated a simulation. That is, I've constructed a set of confidence intervals that could have arisen from randomized trials. I have 20 intervals. The red ones come from trials in which I'm imagining I have enforced balance. And by enforcing balance, I actually reduced the variability in my sample because I've eliminated one source of random variation by enforcing a balance with respect to important covariates. The black represent trials in which I have not enforced balance, and therefore I have more variability, and therefore the black intervals are wider. I'd also add here, I don't know if it's in my slides. Yeah, so balance accounts for one source of variability, so the red intervals are shorter, but the differences here are dramatically exaggerated relative to what you'd ever see in practice. I've never seen intervals that are this much shorter in a real trial when it's balanced versus when it's not. But these are all valid confidence intervals. And to be valid, that means that 95% of the time, they would include the correct value. The blacks include the correct value 95% of the time. We have one that doesn't out of my 20. The reds include the correct value 95% of the time, although I actually have two that don't in this particular sample. That's just by chance or by design, I can't remember. But they're all valid. So they all constitute valid inference regarding the causal effect of the treatment. Balance gains be nothing with respect to valid causal inference. And it's not possible from statistical analysis to determine whether any given CI actually contains the true value. So if I look at this red one here, it misses the true value, but I have no way of knowing that from statistical analysis. It looks just as valid as one that does contain the correct value. So balance may help improve efficiency, but it does not improve the validity of the CI. Attention to tree principle. And I'm saying this is a poorly named idea. It states that all subjects meeting admission criteria and subsequently randomized should be counted in the originally assigned treatment groups without regard to deviations from assigned treatment. Now, the name implies that the analysis is based on the intent of the investigator to treat the subjects in a particular way and ignores whether the subject actually adhered to their assigned treatment. And it's sometimes considered an analysis of the real world effect because in the real world, the subjects, the patients who are being treated don't necessarily comply with the treatment that's been assigned to them. But more important, ITT aligns with the randomization. And therefore it's not subject to confounding, as I've already claimed, and hence it's usually the only analysis that has a direct causal interpretation. So rather than calling it the attention to treat principle, I would prefer if it were called something that had the word causal in it. This is the causal analysis, the randomization-based analysis, some term that reflected the fact that it's the most important analysis that preserves the ability of our trial to provide causal information. And in particular, Dave and I, in our paper, we note that the most commonly used alternatives to ITT actually reintroduce confounding that the randomization was intended to eliminate. And let me show some examples of that. This is one of the most commonly used examples of the problem with trying to account for adherence to assigned treatment. So the Coronary Drug Project, people were enrolled in the late 60s, 8,000 of them, men, because in those days there was this belief somehow that enrolling women in trials would compromise their integrity. That just seems silly in our culture today. 5 treatment groups plus a placebo, randomized 2 to 5, so that 1/3 of them got placebo. It ran about 8.5 years, and the primary outcome was all-cause mortality. So Paul Kanner, who was at the NIH at the time and was a statistician involved in this trial, did some analysis after the fact in which he looked at adherence to clofebrate, which was one of the 5 active arms, noted that some people were less than 80% adherence, meaning they took less than 80% of what they were assigned. It was about a third in each group. About 2/3 in each group received at least 80% of what they were assigned. But if you look at survival based on adherence to assigned treatment, those who received less than 80% had about a 75% survival rate. Those who adhered at least 80% had about an 85% survival rate. So you might think, oh, this difference is due to the fact that not taking your clofebrate increased your risk of mortality. In the right-hand panel, by the way, this is the overall difference independent of adherence to assigned treatment. But if we look at the same result in the placebo arm, we see actually an even bigger difference. So if you attribute the difference we see in the clofebrate arm to the benefit of clofebrate, you must attribute the difference in the placebo arm to the benefit of the placebo. So it's very clear that adherence here is not dictating one's overall survival. It's likely the other way around, that people who are at highest risk for mortality are those who are less likely to actually receive the treatment that you've been assigned. If we look at the ITT analysis, the difference between the treatment arms is essentially 0. So clearly the lower survival among the non-adherers is not a causal effect, rather it's almost certain that the reduced dose is because the subjects at higher risk. If we were to perform an adherers-only analysis, that is, we look at the people who took at least 80%, we would need to assume that there are no other systematic differences, that is, confounding between the placebo and the clofebrate adherers. That is, when I subset to the people who are adherers, then I'm at risk of reintroducing the very confounding that the randomization was intended to eliminate, and so the randomization no longer can help us. Furthermore, since I'm willing to assume that adherence to placebo has no effect on survival, maybe a more logical adherers-only analysis might consider all the placebos against the high dose. So there we would be comparing the high dose in the clofebrate arm against all of the placebos, and there you clearly have confounding. This all placebos contains both high risk and low risk people, whereas the high adherers among the clofebrate people has a higher proportion of low risk people. So that's clearly confounded. Here's another example. This is lumbar disc surgery. Subjects were enrolled with intervertebral disc herniation, and they had a choice either to be enrolled in an observational cohort where they got to select their treatment, either surgical or non-surgical, or they could be in a randomized cohort where they were randomized to either surgery or non-operative care. These are the overall results by ITT, so by randomized group. And among the bodily pain scores, everybody got better. And here I'm assuming that bodily pain scores, higher is better, unlike most pain scores that I'm aware of where higher is lower. But there's no difference overall in bodily pain. There's no difference overall in physical function. Everybody gets better regardless of which treatment group they were assigned to. And again, no difference overall in this Oswestry disability index. But then the investigators, they looked at adherence to their assigned treatment. And they say that at 6 weeks, only 32% of those assigned surgery actually got it. By 6 months, it was 57. By a year, it was about 60%. But among those assigned non-surgery, by 6 weeks, 18% had opted to get the surgery. By 6 months, it was 40%. By a year, it was 43%. And then they said, well, let's reassign people to the treatment that they actually received. So they did a so-called as-treated analysis. And you see these huge differences in these scores when you look at the treatment they actually received relative to what we saw in the ITT analysis. So here I've gone back and I've taken that same figure looking at bodily pain and I've added to it this bar that shows the difference as treated, which is dramatically larger than the ITT difference. But it's about comparable to what they saw in the observational cohort, where people were allowed to select the treatment that they received. But clearly, this observational cohort analysis is confounded because, if nothing else, we see that at baseline, those who got the surgery started at a much lower level or high degree of pain relative to the people who selected the medical therapy. So what do we conclude from this? Well, my conclusion is that this randomized trial is fatally flawed. So to this day, I have no idea, at least based upon this particular trial, whether surgery is better than non-operative care. Everest, this was a heart failure trial. And here what I've done is I've plotted cumulative mortality for over about 2 years. This is Kaplan-Meier estimate of cumulative mortality. But there's one erroneous school of thought that says that events after treatment discontinuation should not be attributed to the treatment. So it's very common, in my experience, that investigators, that pharmaceutical companies, they want to stop counting events that happen after one has discontinued from their treatment. So here I've added a Kaplan-Meier estimate that ignores events that follow discontinuation from assigned treatment. So as soon as you discontinue treatment, your follow-up is censored at that time. We see dramatically lower Kaplan-Meier estimate for the on-treatment analysis versus the ITT. Of course, they usually don't stop the follow-up immediately after treatment discontinuation. There's a window. So this is where I add a window of 30 days, saying we'll only count events that happen within 30 days of treatment discontinuation. This is closer to the ITT. Or we could even go farther out. Here are two curves that correspond to 60 or 180 days. And of course, if I extended that follow-up interval long enough, eventually it would coincide perfectly with the ITT version. Now the kicker is that this is the placebo group. So if you believe that somehow when people discontinue their treatment, that their mortality risk goes up, because when I don't include the things that happened after treatment discontinuation, we see much lower risk, then what did I say? Then you have to believe that placebo is effective at reducing mortality. I don't think we believe that. I think what we believe is this is another instance of the tail wagging the dog, that the people discontinue treatment because they're at risk for death and not the other way around. And furthermore, other than the ITT version, none of these Kaplan-Meier estimates correspond to probabilities in our universe. If you do the math, you realize that this red curve actually doesn't represent any quantity that has any meaning in our universe. So these analyses that try to account for non-adherence cannot possibly fix the problem of estimating the treatment effect among people who actually adhered to the treatment. Here's my final example. This is polyheme. It's a blood substitute given to people in a context where whole blood or typed blood is not available. The conventional wisdom is that non-adherence actually shrinks the observed difference between groups towards zero, and that alternatives to ITT are less conservative. But in this particular case, the first row, which isn't quite IT, but it's the closest thing that we have in this particular paper, the difference between arms is much larger than the difference between arms when you look at either as treated, meaning we switch people into the group based on what they actually received, or the so-called per protocol, in which you exclude people who didn't receive the treatment that they were expected to have. So the conventional wisdom that says that ITT is more conservative in the sense that it shrinks the observed difference towards zero is simply false in this particular example. And because we know that the other alternatives reintroduce confounding, they have no credibility with respect to inferring causation. So causal thinking. ITT analysis should always be the primary analysis because it's the one that allows us to make causal claims. The question that the trial addresses is not, is there a difference in average responses between those receiving treatment A versus treatment B? If that were the question, we wouldn't maintain the randomization. We would actually look at treatments actually received. Rather, the question is there a causal association between treatment and response? So we need to do an analysis that actually addresses the question of causation. Then the common alternatives to ITT, either the per protocol analysis, which only looks at adherers, the as treated, which actually looks at the treatment actually received, or the on treatment that excludes events following discontinuation. They have no hope of actually fixing this problem of non-adherence. So what Dave and I said in our paper, it's unrealistic to imagine that simple statistical maneuvers like excluding non-adherers or switching groups can transform the trial that was conducted into the trial that ideally could have been conducted. Okay, so moving on, what's the question? There are two kinds of questions that we want to answer in clinical trials. The first question, does treatment A improve health state of the individuals relative to treatment B? This is a hypothesis testing question. So just simply answering the question, yes or no, is there evidence that the treatment B is better than treatment A? Now, when I say hypothesis testing, I'm not simply saying yes or no. I'm saying we also have to report the strength of the evidence via a confidence interval or a p-value or some measure that actually allows us to quantify the difference. So we would know whether it's sort of a marginal result or whether it's a very strong result. The second question is, how much is health improved? And this is an estimation question. And this requires that we explicitly quantify the observed differences, usually using one or a small number of parameters. Now, we design trials to test hypotheses. The outcomes are generally chosen to reflect the expected benefit with respect to the specific disease under study. Our sample size and power, primary analyses, et cetera, they're almost always formulated within a hypothesis testing framework. So we never say, oh, I want to be able to estimate the treatment effect with a certain amount of precision. I almost never see that in a trial. It's always, I want to maintain a certain amount of power for projecting the null hypothesis and at a certain level of statistical significance. So except in some special circumstances, such trials may not actually be able to reliably ascertain sort of the net quantitative benefit of the treatment. That is, estimating the so-called treatment effect, assuming that such a quantity exists. And if you would take my class, you would see lots of examples where I argue that maybe this thing we call the treatment effect doesn't actually exist. One example in heart failure trials, it's common to use a functional measure such as 6-minute walk distance to capture the impact on heart failure or the impact of the heart failure on physical function. On the other hand, heart failure patients are also at risk of death. So how do we simultaneously account for the fact that some people die and some people might have improved physical function? Well, I construct a score that says if a subject dies, so I'll give them some very small number like -1,000. Otherwise, I'll use the change in their 6-minute walk distance if they're still alive. But it's unclear in this setting whether there's any single parameter that can capture the net effect of treatment when I have this composite of death or change in function. On the other hand, it allows us to order people by whether they're alive or dead and whether they have improved or less improved a 6-minute walk distance. So I can easily test the hypothesis to ascertain whether there is benefit. It's much more difficult to ascertain the quantitatively what that benefit is. So this comes from a DMC report that we made about 20 years ago with the heart failure study, enoxaparin versus placebo. And here we chose to use the difference of the median. So it's exactly the outcome that I just described. So the median is the median 6-minute walk distance. So it's actually a distance, but it accounts for the fact that people who are dead get these really low scores. So it's not clear to me how one would, in practice, interpret this median, because it could be driven entirely by differences in mortality and no causal effect whatsoever on physical function. So here we see that There is evidence of benefit, but it's not clear exactly how that benefit is manifested, if it's with respect to mortality or with respect to improved function. The same data shown in the final publication, and here they're using the so-called Hodges-Lehmann estimating, which again, it's this kind of non-parametric version of a location shift model that's based upon the Wilcoxon rank sum. And the reason that all these dots are on the right-hand side of the vertical line rather than these which are on the left is simply the convention that this says the maximum better is on the right, and in ours it was on the left. But this is simply another way of quantifying, but again, it's not clear to me that it's easy to interpret these differences quantitatively. Here's another example. This is in polycystic kidney disease. We're looking at percent changes over time in kidney volume. So there's a baseline measure, and then they have MRI scans annually to ascertain kidney volume. And polycystic kidney disease is a disease in which your kidneys develop cysts that grow both in size and in number. But most of the volume of these cysts is water. And tolvaptan is an aquaretic, so it sucks water out of people like nothing I've ever seen before. It's much more effective at removing fluids than is the standard loop diuretics, for example. So what they've shown here is that the average change in the placebo arm was greater than the average change over time in the tolvaptan arm. And you could quantify this difference by saying it's about 2.7% per year. P-value is really, really small. Now, in the end, the FDA decided that they didn't like this as a primary outcome, and they forced the sponsor to do a second trial where they were looking at EGFR instead of kidney volume. But the problem with this figure is that it doesn't actually capture what's going on under the hood. This is what's going on under the hood. But within the first year, the subjects receiving tolvaptan actually had a reduced kidney volume, largely because we just sucked a lot of fluid out of them. But once we've sucked all that fluid out of them and sort of maintained that reduced fluid volume by continuing the dose over time, their kidney volume still increases. And here I've superimposed the means from the primary analysis that was used in the paper against what's actually going on under the hood. And I would argue that there is no single parameter that captures what we're actually seeing with respect to these changes over time. So even though we can say, yeah, tolvaptan has a causal effect on reducing kidney volume, how much it reduces kidney volume isn't clear. Furthermore, there's a question of generalizability. So we have a target patient population way over here on the left. But then in our trial, we say we want to restrict it to people who satisfy certain entry criteria. And these entry criteria may exclude people who otherwise might be eligible for treatment. But then we actually enroll people in our trial who may be just a subset of those who satisfy the entry criteria. And finally, we get a trial result that might well be applicable to the subjects who are actually enrolled in the trial. But the further we go back from right to left, the less likely it is for our result in the trial to be immediately applicable to that population. So I think it's plausible that if I show benefit with respect to the subjects enrolled, that it's also beneficial within the overall target population. It's less plausible to me that the effect size within the trial population will apply to the target population. So I don't take these effect sizes that we might estimate in the trial population too seriously. Here's some text that came from the ICH-E9 guidance. This is the International Conference on Harmonization of Statistical Principles in Clinical Trials. And they say this: by the time confirmatory trials are undertaken, subjects in the trials should more closely mirror the target population. So they want to say these protocol entry criteria should look as close as possible to the overall target population. In these trials, this generally helps to relax the inclusion and exclusion criteria as much as possible within the target population while maintaining sufficient homogeneity to permit estimation of treatment effects. So the idea is if I have a very homogeneous population, I have less variability, I can more precisely estimate some number. If I have a heterogeneous population, I have more variability, and the precision of my estimate is less. Now, in my opinion, this statement is incoherent. If populations are in fact homogeneous, then any subpopulation is representative in some way. I can just select the population that I want as restrictively as possible, and it'll generalize. Of course, I can't know that. On the other hand, if populations are heterogeneous, then the whole target population is not representative of individuals within the population. And the only way to obtain precise estimates is either to do multiple trials in different subpopulations or do one extremely large trial in a wider population, large enough so that I can ascertain whether there are differences in benefit over the range of people in the population. So I actually copied this from a presentation that I saw one time. This is the so-called average patient. But the average patient is actually, in this case, I'm imagining someone used some imaging software to actually literally average all of the people around here. So the average patient doesn't actually reflect any single person within our very heterogeneous population. So what's this issue about homogeneity versus generalizability? So suppose I have a very heterogeneous population, and in my left-hand panel, I see a wide variability, both in the control arms equals 0, and in the active arms equals 1, and I have some mean difference between them. And my target estimation procedure is to estimate this mean difference. On the other hand, if I observe some covariate W at baseline that's highly associated with the outcome, then for any given value of W, I have far less variability, and so I can get a more precise estimate of the difference by looking at people restricted to particular values of W. On the other hand, I can actually fit a model, and the model will have variability which is similar to that of the conditional model rather than the unconditional one. So I can actually recover my precision by simply modeling the relationship between W and Y and accounting for differences between treatment arms. But suppose that we were to enroll just a homogeneous subset in the middle. So I have a narrow subset of W's. I reduce the marginal variation in my lines, so I can get, for the same sample size, I can get a more precise estimate of the overall difference. But if the model is right, and so these lines are in fact parallel, then this subpopulation, this narrow population, is actually representative of the whole population, so the estimate I get applies to everyone regardless of their underlying W. But of course I can't know that because I haven't observed people outside of that range. But the lack of generalizability arises when there's an interaction. That is, the mean difference changes over the population. So over here for small values of W, I have small differences. For large values of W, I have large differences. So the mean difference for the population in the middle does not apply to the entire population. On the other hand, if I'd enrolled my heterogeneous population, I still have the same overall population average difference. By adjusting for W in the way we did before, I can recover the same precision. So if I can select people based on W, that is, I can select by observing W and enrolling only people in the middle, then I also know it so I can adjust for it. So enrolling a more heterogeneous population does not incur a loss of precision. Now the problem is that the average difference does not apply to the entire population, and unless I've increased the sample size dramatically, we don't have the power to know that in fact there's an interaction. So we can't know that for people with small W's, the true difference is small, and for people with large W's, the true difference is large. I can't know that. So what I'll end up with is the overall population average effect, but I don't know whether it's generalizable or not. So the issue of homogeneity versus generalizability, I think is a bogus one. Okay, so that's my little brief discussion of the question. Finally, missing data. I think missing data is the biggest problem that we face in clinical trials. Proper ITT analysis is only possible if we have complete ascertainment of the outcomes of interest for all randomized subjects. And per the article that Dave and I wrote, if I exclude people with missing responses, we reintroduce confounding. So therefore, all efforts should be made to ensure that all randomized subjects are followed regardless of their adherence to the assigned treatment. So back 15 years ago, the National Research Council issued this report on the prevention and treatment of missing data in clinical trials. And one of the recommendations says, trial sponsors should continue to collect information on key outcomes on participants who discontinue their protocol-specified intervention in the course of the study, except in those cases for which a compelling cost-benefit analysis argues otherwise. And this information should be recorded and used in the analysis. Now, when they say compelling cost-benefit analysis argues otherwise, I say, well, if the result of having missingness is that your trial is completely failed, you can't actually draw the conclusions that you wanted to draw, then that's a pretty high cost. And so to avoid having to pay that cost, you either don't do the trial or you make extraordinary efforts to reduce your missingness. So in my opinion, the only analytic solution to the missing data problem is sensitivity analysis. So our primary analysis requires that we make some assumption regarding the nature of the missingness. I have a primary analysis, I make assumptions, I do the analysis, I get a result. Sensitivity analyses relax those assumptions and assess the robustness of the result, the plausible deviations from these assumptions. Of course, here the problem now is defining what's plausible. I need to somehow know what my underlying missingness data mechanism is. And because we never get to observe the missing observations of any trial that's ever been done in history, we can never reliably know what these mechanisms are. So this is a formulation that I'm published in a paper in Stat Medicine a few years ago for 2x2 tables. So the setup is I have a 2x3 table where I observe a certain number of deaths in each arm. I have a certain number of missing observations in each arm. And then I have my total number of subjects enrolled in each arm. So the missingness model here is defined by two quantities. And in this 2x2 case makes things quite simple. One for each treatment arm. So I have a probability of being missing given that someone is dead divided by the probability of missing if they're alive. If the R's are both 1, then these observations are what we call missing at random. And therefore, our complete case analysis is actually perfectly valid from a causal point of view. And deviations from R equals 1 represent alternative missingness models. So skipping all of the details, of which there are many, I have this picture. So the center point corresponds to both R's being equal to 1. This is my missing at random assumption. The further you get away from the center, the further you get from the missing in this the missing at random assumption. And here I'm imagining I have two people. One of them is kind of optimistic, and they have a region that says the values of the R's that are within this dark gray ellipse are plausible. Anything outside of that's not plausible. The skeptic, who might be me, says, no, I think that values farther away are plausible. And then we say, well, given these plausible values of the R's, how robust is the result that we see? So here's an example in which I have 100 people per group. I have a chi-squared z-value of 2.8, corresponding to a p-value of 0.05, and I have about 5.5% missingness. The sensitivity analysis allows us to calculate what the z would be for different values of these R's. And in this case, the magic number of 1.96 is what we would use for a threshold of a 5% type 1 error rate. It isn't even on the picture. So both the skeptic and the optimist would say this result is robust. Suppose I increase my sample size to 1,000, and I have exactly the same z and the same p-value, and the same amount of missingness. Now I see that my magic line that corresponds to 1.96 is starting to get close to these plausible zones. So the skeptic says, oh, you're still good, but not by much. And the optimist says, oh, you're actually still pretty good. Now, if I increase the sample size to 10,000, no one would ever do this trial, but suppose you did, and you saw exactly the same z and the same p-value and a 5% missingness, both the pessimist and the skeptic and the optimist would say, sorry, game over, you lose, because the result is not robust. Here we have 10% missingness and 100 per group, same Z and same P. Both are robust with respect to the skeptic and the optimist. If we increase our sample size to 1,000 and see the same Z and same P, the skeptic says, oh, you lose, and the optimist says, wipes his brow and says, oh, we just barely won. At 10,000 people, it's game over. Here's 15% missingness, but the missingness, missingness rates differ by treatment in this particular example. They're both robust. If we increase the sample sizes again, we lose. And if we increase the sample size again, we lose big time. So the conclusions here, and I have to wrap up because I'm almost out of time. Missing data compromises our ability to conduct valid causal analysis. And surprisingly, this interference can be more pronounced in large trials than in small trials. In some settings, even small amounts of missing is completely sabotage an otherwise well-designed trial. So my conclusion here, my big bottom line is the prospect of missing data in randomized trials should terrify sponsors, investigators, regulators, consumers, etc. And finally, the usual sample size adjustments that we make are likely inadequate to properly account for non-random missingness. So I think I will stop there. Is there an opportunity for questions, or what's the
Yes,
the
there
goal
is.
here?
And we do have some questions already. So I'll start off with Kelly Miller asked a question. What year was the polycystic kidney study done? And was the EGFR corrected for race? She comments, seems challenging to interpret results when the early studies didn't correct. Then there was a movement to correct, and I believe as of late, they want the correction removed again. So can you comment on all that?
That's probably beyond my expertise. I can tell you that the study was, well, actually there were two studies done in the early 2010s, if I remember correctly. I think the last one finished roughly 10 years ago. And I can't remember which formula was used. PKD epi maybe for the EGFR. The sponsor eventually did get approval from the FDA on the basis of EGFR. Now, if it were up to me and I had the disease and I was 30 years old looking forward to having kidney failure in 30 or 40 years, given what I see with respect to the side effects of the treatment, I don't think I would take it. I think the benefit is minimal, and it's not clear whether it persists over the 30 or 40 years before I finally end up with end-stage kidney disease.
Okay, are there other questions from our audience? Otherwise, I can ask some questions, just some very basic questions. So if you are a clinician and you are thinking about launching your clinical trial, at what point should you reach out to a biostatistician? That's the first part of my question. And the second part of my question is, how often would your biostatistician prefer to meet with you throughout the trial?
As soon as possible, because I remember being involved in the very early stages of trial design, and very quickly it was clear that the size of the trial that was required was just not feasible. And so the sponsors ultimately decided, no, we can't even do this, so let's not even bother to go further. So I think it's important to have someone on board early enough to be able to assess whether the trial can even be done. And then I think they need to be involved all the way along, because there are lots of issues that come up during the course of a trial that require the expertise of an experienced statistician.
Very helpful. Thank you. Dr. Carlson has her hand raised. Can you unmute and ask your question, please?
Yeah, thank you. I couldn't type fast enough. Thank you for your presentation. I guess my question was in common in Alzheimer's disease clinical trials that they'll have a cut point, an upper age limit cut point. And so that introduces all kinds of problems in that, you know, they'll have an upper age limit cut point of like 75 or 80. And that's really when we see the prevalence of Alzheimer's and rate of decline accelerate. So I was wondering if you could comment, thinking about representativeness versus, endpoints with, death, lack of missing data, all these things. Can you comment on trials, especially when there's a disease that correlates with age, on having upper age limit cut points in some of these trials?
Well, that's pretty much every disease I've encountered almost. Their heart failure, clearly cancer, clearly they get the increasing prevalence with age. I don't know if there's a rule. I would argue that whatever the context, you need to be able to account for mortality. Unless it's a young population who's not at risk for death. In the PKD study, I don't think anyone died during the trial because they were all early enough in the progression of the disease that EDSR, end stage, didn't happen until much, many years later. So there are trials in which it's not a factor, but if it is, one needs to account for it. Now, I think, at least my father's death certificate, cause of death is listed as Alzheimer's. So I don't know exactly what the biological mechanism is by which Alzheimer's causes death, or if that was just because he happened to be suffering from it at the time. But if there's certainly a causal link or a causal pathway by which Alzheimer's causes death, then I think a trial in older people would be certainly feasible as long as you're willing to account for the fact that these people die. And it's also very difficult to tease out the causal effects on cause of death. Is it Alzheimer's? Is it pneumonia? Is it heart failure? Is it something else? Those are very difficult questions. I know the heart failure people are always wanting to look at CV mortality rather than mortality from other causes. And I try to discourage that just because you avoid the problems with competing risks, which I didn't talk about.
Yeah, I think that's what I was getting at, the competing risks. So it's more so to look at the outcomes are not necessarily death, but it's a cognitive outcome or biomarker outcome. But I think the sponsors are afraid to enroll with older people.
Yeah, I get that. And these are hard. But one of the semesters I taught my class, I ran out of time to do anything substantive at the end. So my final slide just said, clinical trials are hard. That was the conclusion of my course.
Any other questions from our audience before we wrap up today?
Yeah, that was a very quick sort of tour through a lot of the issues that I deal with in my class and in my real life.
Okay, so I'm not seeing any other questions. So again, Dr. Cook, thank you so much for presenting on biostatistics for clinical trials. We really appreciate it. This recording will be sent out a little later this week along with some additional materials. And thank you everyone for joining us. We appreciate it. Have a good day.
Yeah, thanks for inviting me.